Good evening, scientists. Today we delve into the fascinating world of genetic screening. Genetic screening is a powerful tool that allows us to analyze an individual's DNA to identify genetic variants associated with or without diseases or other traits. This information can be crucial in detecting, preventing, or managing the genetic disorders, as well as informing medical decisions. This is formed by Yannick, Gurraj, Kushmi, Obritiy, and Amjad.
In this slide, I will talk about what is genetic screening. It is the process through which we can check for and find changes in DNA in an organism. Genetic screening can identify gene mutations, chromosomal abnormalities, or variations associated with disease risk. This information can help in diagnosis, treatment, and prevention of genetic disorders. We will discuss this further in the next slide.
We're going to delve into the two examples of forward screening and reverse screening. So what is forward screening? A method used to identify the genetic basis of a phenotype without prior knowledge of the gene involved. And the processes: mutagenesis, inducing random mutations in an organism's genome; phenotypic screening, observing the resulting phenotypes to identify interesting traits; mapping and cloning, determining the genomic location of the loci and isolating the gene responsible for the phenotype. A few examples: researchers randomly induce mutations in a population of pink flowers. They then observe a small sample of white flowers and deduce that the production of pink pigment is controlled by the gene. Let's move on to reverse screening. Reverse screening begins with the sequence of a gene and works to determine its function by mutating it. For example, researchers begin with a known gene and then look into the phenotypic changes that occur when the gene is tweaked or deactivated. Another example is when a gene knockout mice, disabling a specific gene on the mouse phenotype.
This is our focus question. You have treated a wild-type fly strain with gamma radiation to induce mutations. Explain how genetic screen for eye color mutants could help verify the randomness of mutations. Objective: verify the randomness of mutations induced by gamma ray radiation. Method: cross wild-type flies with gamma irradiated flies, screen offspring for eye color mutants, analyze mutants for diverse eye color phenotypes. Controls: untreated flies as controls, known eye color mutants as positive controls. Analysis: genetic mapping and complementation tests to confirm mutations. In conclusion, a genetic screen for eye color mutants in fly can serve as a valuable tool in verifying the randomness of mutation induced by gamma ray radiation. This approach provides insights into the mutagenic effects of gamma radiation and the distribution of mutations in the genome.